forward. Hello, everyone, to our September 2024 Symbiota support group. Let me share my screen. Thanks for bearing with us and joining us a week later than previously scheduled. Thanks to the Spinach-Tadwig joint meeting last week. We're really looking forward to talking with you all about today's topic. But first, I'm going to hand it over to Lindsay to do some announcements.
Awesome. Thank you, Katie. And I think next month we're going back to the normal programming for Symbiota Support Group.
Yes, good point. So that one's going to be on the October 7th. So mark your calendars for back to our normal.
Yes. And so to continue from last month's Symbiota Support Group, this month we're still talking about Symbiota 3.1, but doing a little bit more of a deep dive, and Katie will talk about that in a moment. But we are continuing to roll out Symbiota 3.1. And so if you missed last month's Symbiota Support Group meeting, we have an entire recording on some of the highlights from Symbiota 3.1. And we also have some blog posts about that on our website if you just want to kind of read through the highlights of it. And then we have another communication that came out recently just about like the rollout process. So there's also a post about that on our website as well. If you missed that, we just wanted to alert everyone to that in case you missed either of those emails or you might have missed the support group meeting last month. And it all relates to what we're talking about today. And then unrelated to 3.1, if you have vertebrate collections, if you're either in CSV Call or maybe you're thinking about it, we're hosting a portal advancement campaign for this portal with Dakota Rousey, the portal manager, starting later this month. And so if you might like more information about that portal community or you're already in that portal, please feel free to join us. We're going to have at least one meeting. It'll be on a Tuesday, I think at the end of September. And so the registration link to that event is posted on our website, and Katie just put a link in the chat to where you can find that information. And then one more quick announcement. You might have seen an email that came through last week about the Symbiota Support Hub transitioning to the University of Kansas. What that really means for you is that not a whole lot should change and that you can still reach us at our regular help desk email, help@symbiota.org. We did send an email out, like I mentioned, about that. So just refer to the email for more information, and there'll be more time for questions if you have any after the presentation.
Yeah, and that email is also pretty much summarized in the link that I've just put into the chat. So if you want more about that, go for it. Okay, let's dive into today's topic, which we referenced last month, but we're going into a deeper dive this month. And that is exploring this new tool that was developed thanks to some funding from the Botanical Research Institute of Texas and the California Botanic Garden and George Mason University. They all had a collaboration that funded this. And that's the ability to batch upload linked resources and associations. So first, we're going to talk about what linked resources and associations are, just so we can wrap our minds around that concept first. And then we're going to talk about how you can link associations to your records, both kind of individually and batch, just so that we can cover all of our bases there. I think we should have enough time. Okay, so what are linked resources or associations? One way you can think about this is in terms of the digital extended specimen or the extended specimen. And this is the general concept that a material specimen, a physical specimen, which you can think of as this part of this diagram, is just one part of the important resource that this specimen provides to science, because you can kind of extend this specimen to include pictures of that specimen and that specimen's digital record. So that's not necessarily an attribute of the physical specimen, it's an extension of the specimen. And then even beyond that primary specimen data, you can think of extending the data even more by linking it to information about the specimen, such as field notes or subsamples of that specimen, any like morphometric data, phenological data, trait data. And then even beyond then, you can think about expanding it into like traits of the species and evaluations of the rarity of that taxon, etc. So the idea is that a linked resource or association is some extension of that specimen's data beyond like where and when the specimen was collected. So it's like more information about the collector who collected that specimen, or more information about the field images or field notes, something like that, that's not intrinsic to the specimen itself. So in Symbiota portals, there are 4 types of linked associations that you can create. And I'm going to walk through these because they're also going to show up in the tools that you use. There are non-occurrence resources, and I'm going to define these later. Occurrences, internal, occurrences, external, and then taxon observations. So I'm going to describe these as I talk about how you link them. Oops. Here we go. Okay, so the first one I want to talk about is occurrences. So an occurrence is some other specimen or observation. Occurrence is like this data standards term that means the instance of a taxon at a place and time, usually collected by a person. And so an occurrence is just something, some taxon that was collected or observed at a place and time. And the 2 different types correspond to whether that occurrence is in the portal that you're linking to, or if it's external, then it's in some other portal. So you've found a specimen record that belongs to, that's in like an Arctos database that is related to your specimen somehow, versus like, hey, my herbarium down the road has a subsample of my specimen, like a piece that was taken out of it and was used for sequencing or something. So some examples can be like host parasite interactions. You could say if someone collected a host and a parasite at the same time and same place, then you could link those 2 associations. Or there could be other vouchered species interactions that you're talking about. So say you collected a specimen and then you collected a specimen that was like a neighbor of that specimen. So like, you know, an oak tree that had some herb underneath. Sorry, I keep on using plant examples, but that's what's on my mind. Then if you collected both of those specimens at the same time, they technically can have an interaction, and that interaction could just be like ecologically occurs with, but that could be an association. Donna asks a question, what about herbarium duplicates? That's a great point, Donna. And Symbiota portals have had the ability to link duplicate specimens for a long time, and that's actually a little bit of a different module in Symbiota portals. For those of you who don't know, duplicate specimens are usually plant specimens that are the same taxon collected at the same place at the same time, either corresponding to the same individual or the same population of a certain taxon. And so those are very intricately linked associations. And those are treated by the kind of the duplicates tools. And Lindsay, if you could put a link to the documentation about duplicates in the chat, that would be helpful. Okay, so this was just kind of a little clarification again, that you can link specimens to occurrences that are in the same portal or a different portal or database. And that's the difference between the 2. So what type of relationships are we talking about? Well, there's a relationship controlled vocabulary that's currently loaded into each portal that has these tools available. And this is subject to change. This could be changed by a community saying like, hey, we have a bunch of this type of resource that we want to link. Can we add a new relationship to this controlled vocabulary? And that's great. But here are some examples. There's like derived from same individual. If you have like multiple subsamples of a single sample, there's ecologically occurs with, there's has host, host of, part of, and then referenced in and references, those probably would refer to non-occurrence resources, which we'll talk about in a second. Ed makes the point, very important point, that exicati are also considered a different specific type of specimen association. And that the exicati has a different module for making these types of associations. And yes, and they're also in, if we go into a record, exicati are also in this linked resources if exicati are available in your portal, which I believe they are not in CCH2. Karen asks, can you discuss multiple species within the same specimen? Yeah, that's a good point, Karen. I'll come back to that, but that's a very good use of this associations module, I think. Okay, so that's one type of association. Let's talk about non-occurrence resources. Those examples would be things that are, again, not an instance of a taxon at a place and time, but are still some sort of related information to that specimen or record that you're looking at. So this could be field notes. This could be a literature citation of your specimen. This could be project or permit information. Now note that you're not allowed to upload a PDF or any other sort of document into this module. You can just put a link to somewhere else you have put that information. So if you have some sort of repository available to you where you can put that information, you can put that link here. And then if you're doing literature citations, you would want to do something like a DOI. Okay, the last type is a taxon observation. And this is an instance that is not vouchered. So that means that you're making a relationship between your record or your specimen and some other taxon, but that taxon doesn't have like a specimen or a specific observation related to it that you can link to. You're just making an kind of a formalized statement in your data that says this thing is associated with this specimen at this place and time via some relationship term that you select. So some examples could be like the flower was visited by blah, blah, blah. Like I've seen this on some specimens where it says like many Bombus visitors. And so you could put an association between your specimen and Bombus. Or you could say, if it said like on an oak tree, like if you have a lichen and it's found on an oak tree, then you could say on and then make the taxon association Quercus. Or parasitized by, or collected off of, or just associated with. Anything that goes into the associated species field could technically go into a taxon observation. Don't start translating all of your individual values in associated taxa into this. We would be able to do that batch at some future time. But just so you know, that's what a taxon observation is. It allows you to link to a taxon using a relationship term instead of just saying, my record was associated with this other species. No really telling how it was associated to it. Donna asks, separate fruit DNA, etc., would count as a subset, right? What about image or photo files? Donna, are you talking about images of those subsamples or images of the specimen?
I just, thanks, Katie. I apologize for interrupting. I just kind of meant the whole specimen images that we're taking and then are obviously kind of linked. They come up when you do the occurrence record. And I also definitely vote on Karen's, the mixed collection nightmare too. Did you hear
Yeah.
about that one?
Great. Yeah, you're not interrupting at all. Like this is, we want questions as we go. The images are already in kind of a different extension. And that's why they're in this images file, just like traits are also an extension, but they're in some separate module because they can be treated more nicely in this other module. So those are also extensions, just a different type of extension treated in here. And then separate fruit DNA, etc. Yeah, I would say separate specimens that are related could be linked in this associations module.
Yeah, so for example, we have large bins of oversized pine cones, some and many of which have a press specimen for needles or leaves for, a big and so maybe that would be a linked resource.
Yeah, depending on, those might be considered duplicate specimens because it might be coming from the same exact population at the same day. So that might be considered a duplicate, which is,
I can go with that. I mean,
yeah.
you just, so it to me, I mean, it could easily be considered, to me, a duplicate is just something that I sent off to another herbarium because there was too much to mount on one sheet. In this case, it was just because, well, that daggone, walnut or pine cone was too big to put on the sheet. So we have a separate box to put it in, but it literally will have the same exact label as the herbarium sheet and the same session number the way we do it.
Yeah, so I would still consider those probably duplicates, but you could instead like make yourself another record that's like, here's my cone record, so that it has like different information about where you physically placed it in your record, in your specimens. Or, and we're not going to go into this in this webinar, but you could list that as a material sample of your specimen. So there's a possibility where you can have your specimen record and there's a different module called material sample. And it allows you to say like, oh, I took 3 DNA extracts and
that's
and here's my cone and here's my whatever else you might have taken from that specimen or is associated with the specimen. So
that was my final interest because we have so many people now sampling and taking DNA vials. Is that also is considered, you said a material sample is the correct terminology for that?
There's a couple of ways that you could link those. There's also a genetics module here, see? So if you have like a link to where that genetic sequence was deposited, and they should always do that, then you can just link that genetic resource directly, and it's going to go into a field called associated sequences, which is slightly different and may be queried in different ways. It's a little bit complicated because there are actually a lot of ways of representing the same thing using different modules, because you're like, well, that's technically a material sample of my specimen, but it's also a sequence, but you could call that an associated resource. So kind of the rule of thumb is try to use the most specific tool to your instance. And then if you can't, find a box to put it in, then use associated occurrences or linked resources. This upper box up here. So if you have like a duplicate, use duplicates. If you have genetic data, use genetic stuff. But if you have a link to something else that you can't fit in those boxes, then use this here. And Ed, I'm just realizing that this says associated occurrences and it should probably say resources because we have non-occurrence resources is also an instance here. But I digress. I'm not showing you how to actually do this. Okay, let me go up to some examples here. So I wanted to make sure that everyone has a link to our documentation, because this can be a little bit challenging to navigate. And we have clearly defined these 4 types of associations in our documentation and provided some examples and provided, where are they? Provided templates. So if you're just overwhelmed about how you're going to batch upload these things, you can just download a template. You can be like, okay, I know I have a bunch of field notes, which are a non-occurrence resource. I'm just going to download this example template and then fill it out. So that's a good way to go forward. And so, yeah, there's different parts for internal, for external, taxon observations, and non-occurrence resources. Okay. What was I doing here? Okay, so let's just go do some examples here. So I'm first going to do a taxon observation example. Say I go to my specimen records and I want to look for anything that might have a taxon relationship that is a parasite relationship. So I'm going to go, here's my collection profile, and I'm going to go export a file that has anything in the occurrence remarks with parasite. I'm gonna leave the E off so that if it says parasitizing or something, you can do that. Okay, I'm gonna get it just a Darwin core output. I'm gonna download my records. And I have a bunch of specimens that might have a parasite. So I'm going to go to my occurrence remarks. And these ones say parasite on. So that's cool. I'm just going to use a subset of these. Let's just do a few. Okay, so where I'm going to do this batch upload of associations is under Administration Control Panel, Import Update Specimen Records, Extended Data Import. And then I have to choose associations from this import type. And I have to upload my file. And one caveat is that you can only do one association type at a time and one relationship at a time. And the reason for this is because, as we mentioned earlier, the relationships in this list are a controlled vocabulary. So these are the only values that you can select from. And again, you can ask to have more added, but this is a drop-down list. If you were uploading a spreadsheet that had multiple different types of relationships, the database has no way of easily controlling the vocabulary that you're using in the relationship. So that means that when you're uploading something, you should upload only things that have one relationship type. So only things that say has host. And you could include that in your spreadsheet just to like keep that on the top of your mind. But just note that there will be a step later in the process where you're going to say, all of the records that I'm uploading right now have the relationship has host or whatever you are using. Okay, so I am going to import an association, and the association type that I'm demoing right now is a taxon observation, because I want to link the fact that my specimens that I'm working with are parasites of whatever taxon is provided in this occurrence remarks field. And the required, let me pull my documentation back up. In all cases, the first required field in your upload spreadsheet is going to be an identifier to the records that you're trying to link to. And that identifier can be a catalog number, an other catalog number, or an occurrence ID for your specimen record. So in here, I'm seeing that some of my specimens don't have catalog numbers. So I'm probably going to want to link on the occurrence ID. So that's going to be my main identifier. And you'll see some verbiage throughout this documentation that uses the words subject versus object. And the subject, it refers to the record that you're adding an association to. So if you were going to just do a single association, I'm going into my record here. You can think of the subject as being this specimen that I'm looking at right now. The object would be whatever you're linking this record to. So that might be just a URL, that could be the object, or it could be another specimen somewhere in your database or in someone else's database, and that would be the object. So that's just kind of like a data standards term of just think subject is the thing you're looking at the screen right now, the thing that you are putting something onto or linking something onto. And in the documentation, it's going to be listed first to kind of help you think about that. So my subject is the specimen that I'm putting the associations onto. And then a taxon observation upload is pretty easy because the only other required field, we need an identifier for the subject, and the only other required field is the scientific name. So all I need to do is extract the scientific names from this remarks field. So I'm going to get rid of some fields that I don't need from this particular download. There's my occurrence remarks. Remove all of these. Okay, and just to help myself remember what type of relationship, I'm going to put relationship type here so that I'm remembering, okay, so the relationship between my subject, which is occurrence ID, and this taxon is has host, because my subject has a host of whatever it is parasitic on. And conversely, if this record was like the host and the occurrence remark said something like parasitized by many aphids, then I would want to say, okay, my subject is the host of whatever the taxonomic name of aphids is. So it's a little bit hard to keep straight sometimes, but if you just put it in a sentence, that's what helps me. I'm like, okay, my record has a host of this taxon. All right, I'm gonna make this smaller just so that we don't spend a lot of time on this. So now we need my one other field, which is going to be scientific name. Actually, I think it might have another name here. Verbatim sign name. And the names of your column headers do not have to perfectly match the documentation. It's just helpful to reference. And I'm gonna do, what I could do is I could individually pull out all these records and say, okay, I'm just gonna say, this one's gonna be Populus Fremontii, put it here. Or you could come up with some script that will do it. I'm going to demo, I'm increasingly using ChatGPT to do things. I'm not necessarily saying that they're the best or that you should use them for everything, but here's just kind of a quick way that I've found. I could say extract scientific names from this column. And I'm gonna take those all here. I'm gonna stick them here. But in this case, it looks like I have 3 scientific names associated with this one taxon. So I'm gonna have to insert some duplicates here for this name and these 2 names. And these are all gonna be has hosts. And then I have my Salix, my Populus, my Sambucus, and my Alnus. So it's a little bit faster than individually typing all those out in this particular case. All right, now I don't need occurrence remarks. And I can just save this. And then come back into my importer. Okay, so I can choose that file. I'm making a taxon observation. I'm going to initialize. And here's where it's asking me what relationship I want to define between my subjects and the scientific names that are in my file. And we already talked about this. This was going to be has host. And you can use a relationship subtype. I don't think there are super helpful subtypes in here right now, but if you have a desire to consistently define a subtype, for example, we could add field notes to this subtype if you have a lot of those to add, then just let us know and we can add that to the controlled vocabulary. Okay, so occurrence ID, that's my subject identifier. I also put catalog number in here, but it's probably not necessary to do that since I have all of my specimens have occurrence IDs. Then my verbatim sign name has been auto mapped to verbatim sign name. And I'm just going to leave this relationship type blank because that was just for my own mental sanity. Okay, so now I'm going to click import data. And it's creating a bunch of associations. So if I open this up, I can go to my linked resources and voila, now I see that this Cuscuta, which is a daughter, it's this weird stringy mess in here, has the host of these 3 taxa. So that's a taxon level relationship. Okay. Now, if you want to do a, well, back to my non-occurrence resource, it's also very similar. Now, let's see if I had an example here. Bringing up my, okay. So this would be if you have, let's say we have a citation that we want to link to our specimen records. So one example that you could think about is if you go to your GBIF citations, if you publish your data to GBIF, you get a big list of all of the citations of your data. And you could theoretically look at the data sets that that researcher used and see the specimens from your records that that data set used and link that literature citation to all those records. And I'll show an example of this. So here are a bunch of citations of the OBI collection. And let's just use this one as an example. Oops, I don't want to go there quite yet. If you click on the DOI, so this is, will show you the data referenced in this work. Click on the DOI. It will show you what the researcher, what query the researcher ran in GBIF. It said, it just looked for everything that was this species. And then you can look for your data set in that list and see that 10 records of yours were cited in that, in this citation. So if I do a search of my data set currently, let's just do it in the public search. And only use my collection. I see that indeed there are 10 specimen records. Okay, so I can use this as the starting point for my template if I want to. So again, I'm going to download. I don't need all this additional stuff because all I really need is like the catalog numbers or the occurrence IDs from this particular, for this upload. Oops, I got a tab delineated. I only wanted comma. Go. Okay, now let's go to our documentation and determine what our required fields are. There are only really two. There is subject identifier. So again, subject is my record, my main record. And then the resource URL that you want linked to that record. So that will probably just be the DOI to this citation. So I'll just be grabbing this DOI. But I'm starting with my little template here. And not all of my specimens have a catalog number. So I'm going to use occurrence ID again. And then I really don't need any of this information because I have my subject identifier. And the other required field is resource URL. So I'm going to put resource URL. That being said, I might want to put more than just the resource URL. You probably want to also add an object identifier of some sort in this case. Al asks, have all the OBI specimens lacking catalog numbers not yet been imaged? Yeah, usually those are somewhere just in the processing pipeline. They're like brand new specimens. All right, so I'm gonna go copy this DOI. I'm putting that here. I'm rolling it down. And then let's also put something in the object identifier. One thing I've noticed that we might actually want to change is that if you don't have an object identifier for your resource URLs, if you go into, once you upload an association, it will show up on the public display page about right here. And it will say linked to, or actually it will have the relationship. It'll say, you know, referenced in, and then it will have the resource URL, but that resource URL will be in, well, I'll just do this and then I'll explain in a second. Okay, so let's not use object identifier. Let's just put my occurrence ID in my resource URL.
Katie, I think your DOIs were missing a number at the end, and I want to make sure it works when you do the demo. I think it's missing a 4 after the 3.
Thank you very much. Good catch. Copy that. All right, save. Okay, so to do this upload, you start in your data editor control panel. You go down to import update, extended data import, choose my file. This is again an associations upload. I'm adding a non-occurrence resource because these are linkages to a literature citation. And the relationship that I'm going to choose is referenced in. And I have my subject identifier correctly linked and my resource URL. Let's just import that data. I got one error because I have already done this before individually. And so it said, hey, you can't add that twice. So there's a nice check there. So if I go to this record and I look at the public display, you'll see that it says referenced in and then nothing. What I was trying to talk about before is that the default is that it will show whatever your object identifier is. And then that object identifier will be a hyperlink to the record, or sorry, to the resource URL. But if there is no object identifier, then it just leaves this empty. So this is probably something that we need to change on our end, that if there is no object identifier, it does just also provide the link there. So it's probably also something we need to put on our to-do list. But if you want to avoid that in the meantime, then I could just go back to my file. Oops, back to my desktop. And add a resource object identifier. And since this is all the same object, it's okay to use the same object identifier. And whatever you want to appear right here is probably what you want to put in that object identifier for now. So I might just say like Maharaj et al. 2024 South African Journal of Botany. And I'm just going to put that here and save. So now I'm going to show you, hopefully if it works, how you can edit existing references or resource links. So if I go back to my extended data importer and I choose the same file, and I do an associations, non-occurrence resource, referenced in, and then my object identifier, the object ID. If you check this box that says update or delete records with matching identifiers, so this is going to be subject identifier plus object ID or resource URL. So thus, it's going to look for any of my shared subject identifiers, so occurrence IDs, plus either the object ID or the resource URL. So because I haven't changed the resource URL between version 1 and 2, it's going to find that resource URL and subject identifier pair and just update the records. So if I click import, it says, yep, association updated. So if I go to that record, I see that linked resource. I see the object identifier that I gave to it. And then if I go to public display, now you'll see that it says referenced in this here. And if I click it, I go to that neat citation here. Walter says linking references could be really useful for type specimens. Yeah, that is a great point, Walter. You can link the URL for the Biodiversity Heritage Library or to Tropicus. Yeah, you could get a link to where that specimen was originally cited and then add that via this, this tool. Okay, I won't walk through the whole shebang for the last resource type, occurrence internal versus occurrence external, but I will show you where to get that information. Again, it is here, and it's going to be under one of these 2 or 3s. And again, you can download a template of either source. And just note that you always need a subject identifier and an object identifier. And so the subject identifier, again, could be a catalog number, other catalog number, or occurrence ID. And then your object identifier can be a catalog number or an OCC ID or an occurrence ID. An OCC ID is, if you go to a record, it's the same as this Symbiota ID. So you can find it either in your table display, or if you do a download, like this one that I did, it's going to be the ID in that ID column, if you're downloading it from a Symbiota portal. And then just remember of the key difference between an internal link and an external link is that if you're using an internal link, then you can use those OCC IDs and those catalog numbers, and it's going to link within your portal. But if you are using an external source, so again, some other database that is not the current portal, then you'll need a resource URL. So it doesn't necessarily need to have this object identifier. It's recommended because then you can have people be able to see those 2 catalog numbers in one place, for example. But we're assuming that the minimum that you'll have is a resource URL, a URL or a link to that record in whatever other database you're referring to. Okay, So one last thing with regard to this is if you're like, I just made a big error and I need to batch delete records, you can also do that through the same exact tool. You can upload your record. And then under these relationship types, you can do batch delete. And it's going to map based on these qualifications. So it's going to look for the same association type and the same record ID, or the same association type and resource URL or object ID. So it looks for like pairs of these. And you can refer back to this in the future. Okay, so that was a little technical. Hopefully that gives you an overview of kind of the possibilities, gets your minds worrying about what you could link. And we only know kind of currently a few use cases of this, but we're interested to hear what you guys want to link. Like, what are you thinking now that you might want to add? I love Walter's note of putting type specimen references. I think that's awesome. Does anyone have any other ideas or questions? This is also our question time.
A comment. Just get people thinking about it. If, say, this tool appeals to you, but you're feeling a little bit shy about it, you could always just try it with like a record or two to see if it works the way you want it to. And likewise for the batch deleting, just do it with like 1 or 2 records just to make sure it's like going the way you planned it to, because I could see how it might be a little intimidating. But yeah, there are quite a few possibilities here.
Definitely. Yeah. It's always like, you're not going to break something by just putting 1 record out there. Just try it. Jennifer says that they have been including iNaturalist links via this linked associations. That would be an awesome use of this. There might be some like consideration about whether that like constitutes a duplicate specimen, but without any other uses, I would say that, or information, I think that would count as like an external occurrence. So that would be a great, like you just put your catalog number and the URL to that iNaturalist observation, and then you've made a solid link between those 2 databases. Is there a way to link 2 records for 1 specimen within an herbarium? For example, a specimen that be considered to be a lichen and a fungus. Karen, are you saying that like you have the record in both your lichen collection and your fungus collection, and you're trying to say that they're the same thing?
No, we have people who think this should be filed within the lichens, it should be database within the lichens, and then other people with the same specimen are like, no, that's actually should be like within the fungi. And so if people want to search for that thing and they're like a mycologist, they're looking in the, fungus database and the reverse. So I just wondered if there was a way of just kind of having a cross-reference so both perspectives can be accommodated, even though there's only just one thing.
Yeah, I think that it depends on probably how your herbarium is laid out, like whether your lichens are separate from your other fungi.
They are.
Okay, so then in that case, I would probably create an additional collection in MycoPortal that is just your lichen collection. So you have your like other fungi and your lichens, and they're in the same portal, but there's 2 separate collections, and one of them you could live manage, and then the lichens one is just a snapshot of your lichen portal database.
Great idea. Thank you.
No problem. And you just reminded me that there was a question that I said I would come back to from you. And that was multiple species within the same specimen. And what I would probably do with that is that's a perfect example of when you'd want to create this linked resource. So say that you have a specimen that is a log, of a certain species and it has a lichen and a bryophyte on it. And you're like, well, shoot, I manage these in 3 different databases. Like I have my lichen portal, my bryophyte portal, and SciNet. What you could do is link those as external linkages. So you could go into linked resources and say, this is a occurrence external, and say this is my log. I would say this is the host of, and then you would put the link to your lichen portal record here. And then you would do that with your bryophyte portal record too. And then you would want to do that reciprocally in your bryophyte and lichen portal records. I think that in the future, there's going to be more streamlined ways of doing this so that you don't have to like continually make all these spreadsheets and do a bunch of importing between them. But if you want to get kind of everything sorted out right now, I think that's your best bet.
Thanks.
Any other questions, ideas? Cool linkages? Kirsten says, I'm looking for a place where I can record associated ethnobotanical information. One problem is that some ceremonial information is not public knowledge and I cannot post it publicly. Yeah, that's a big concern, is sometimes you have data that you want to link that's not public. You could put associated ethnobotanical information, you could link it as an associated resource, but then wherever that link goes could be a protected site. So it might like require a login to get into if you just kind of want that for your own internal methods. But I would think long and hard about why and how you want that information to be linked to your specimen records. If it's just for internal use, then maybe you should manage it internally, because things on Symbiota portals are inherently public. So. Other questions, ideas? All right, well, thanks for joining us today. Oh, wait, Nadia says, we do ecological inventories in which we collect specimens of most plant species within a polygon over several days. Would it make sense to link these specimens as ecologically occurs with? We might revisit an area that was surveyed decades ago. Should we link specimens from the same site separated by decades? I don't think I would link those because they're not happening in the same time, and they could be linked by the location in the future. You might consider investigating the field location ID and providing a unique identifier to your locations. And that way they could be kind of linked in an automated fashion in the future. And feel free to send us an email about that and we could point you to some more resources talking about that. But that's a great question. All right, well, thanks again, everyone, for joining us. As I mentioned at the beginning, our next Symbiota support group is going to be October 7th. And have we announced we have not announced what that subject is going to be. So stay tuned, look at your emails, and we'll let you know when we have that finalized. Thanks for tuning in. Send us all your questions about when you use this tool.